All right. So I think I'll be recording now. Yeah. It'll be a little icon there. Recording on this computer, and I have 2 o'clock. There we go. So good afternoon, Anjali, Diane, Diana, Katachi, Felicia, moving up and down, Kezi, and Swinaila. Hello. Good
Good evening.
afternoon. Good
Good
evening. Good
afternoon,
evening.
Good
sir.
afternoon
Okay,
to
great. So
afternoon.
let's
Okay.
excellent. So we have 2 o'clock. I know you're all about business, so let's go. So in terms of the nervous system part 2, what we will be looking at today is the somatic and special senses. All right, so that part of the nervous system associated with those things. So let's go, shall we? Okay. So in terms of the nerve, one of the special things to take note of when we mention the somatic and special senses and senses in general, we mentioned last day, which was when we were referring to the nervous system, how it keeps us in touch with our external environment. Interestingly, when we look at the different senses, and by extension, how many senses do we have? I forgot. Somebody want to help me? How many senses do we have? Not all at once.
5.
5 senses. What are they? Anybody want
Sight,
to pipe in? Sight,
taste,
taste,
smell,
smell,
hearing, and
hearing,
touch.
and touch. Yeah. Some people will want to argue that there's a sixth sense, in which you could perceive things that are not, like perceive things which are just not seen or which the other 5 senses do not pick up on. I might have mentioned it to you. I have a colleague over at Emory University. He's doing research on it. He's a neuroscientist, and it is believed to be true that in terms of the workings of the pineal gland, um, that there is this ability, some people do have it, to detect things outside of the realm of the 5 senses. And interestingly enough, it is located very just between the eyes and a little superior to that region. So we do know in some traditions, particularly in the Hindu tradition, you might see they speak of this third eye. And other traditions also speak about this ability, to see sight beyond sight, as it were. So there does seem to there is some truth to it. And I'll really try before the end of the semester, which is the next couple of weeks. Let me see if I could get a Zoom call to my friend um, over at Emory for him to talk a little bit about his research, which is rather fascinating, I believe. Yeah? So, all right, so we'll see how that goes. Okay, so notwithstanding that sixth sense, which I mentioned, associated with the pineal gland, but they are normally, in terms of what the literature will tell us, there are 5 senses. All right. So when we think of these 5 senses, it is important to note that these 5 senses exist because we perceive them in the brain. So that's very important. So we could argue then that, do these things really truly exist? Well, it depends on your slant to it, because they only exist insofar as your brain perceives it. What do I mean by that? When you smell something, is there a direct channel between, let's say, a doubles? Is there a direct channel then between your nose and going up into your brain? What do you say about that? Let me hear. Is there a direct channel going from your nose up to your brain? Anybody? Is there?
Yes.
Is there, like, so in other words, then the smell then is go up your nose and through this little channel like a tube up into your brain? Yes? What do you think? Yes? Well, the answer is actually no. It doesn't happen, man. It's the same thing with all your senses. When you touch something, right? Let's say you touch something hot. Does the heat travel then up your hand in terms of, ow, so it burning all the way as it go up? and it go all the way up to your brain, and when your brain get burned, you feel your ball, aye, aye, aye. And the answer is actually no. And with all of these senses, one of the things which we will realize or come to realize as we go through this chapter is the fact that all of these senses that or the things that we perceive, they're actually done through the workings of or the sensitization of receptors and the sending of a signal, right? The signal is sent along the nerve and it goes to the brain where it is perceived. The brain then actually draws reference to something that is similar to it in terms of what is stored in memory. And after drawing reference, that's how you're able to identify it. So when we smell a mango, how do we know it's a mango? Well, the sensory receptors in your nose, right? You smell it, and those chemical messengers that come off the mango, what do? You have these chemical messengers, little chemicals. They actually bind to specific receptors in your nose. When they bind, they initiate action potentials. We saw action potentials earlier when we were talking about the neuromuscular junction in terms of the binding of acetylcholine, that neurotransmitter. And in a similar way, so when the chemical odorants actually come off the mango, they bind to receptors in your nose, and that initiates a signal, not just one signal, but several signals that actually go to the brain. And because of that particular combination of signals, when it reaches your brain draws reference to it from memory, and that's how it knows it's a mango. So a mango to your brain might look like this. Bzz, 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 in terms of signals going to it, electrical impulses, action potentials. That's all the brain recognizes, action potential. And it's the same thing when you look at the other senses. When you see something, what happens? You have receptors, and when the receptors, when you have are excited or certain things bind to the receptors, it initiates a cascade of action potentials. And these action potentials are electric signals. They are perceived by the brain. The brain only recognizes these electrical signals, nothing else. So one could argue quite rightly. So let's say somebody who's working on a high tension wire, and or let's say if you are adventurous and you go and try to figure out the workings of a socket and you push your finger in it, the possibility exists if you do trigger the right receptors, right? Yeah, you could smell pineapples, you can have that crossover. If the correct action potential signals are sent to the brain, it will indeed be perceived, as, let's say, what is in memory as the smell of something such as pineapple. So one of the things before we start this session, in terms of the senses, do take note of the fact the brain only recognizes action potentials and electrical signals, nothing else. Okay? We good? All right. Okay, let's go. Oops. So in terms of sensing, the terms associated with the sensations or the five senses that we mentioned, you have sensing, sensation, and these receptors. Sensing, of course, as seen here, is the detection. The sensation is a conscious perception. So for instance, when you sense that a mosquito has landed on the back of your neck, what you'll do, you'll quickly take your hand and smack the back of your neck to send the mosquito to the other side, as it were, right? So that's the sensation. And not only after you slap it, you do feel the sensation of a lash. Depending on how heavy your hand is, you will feel that sensation, right? Most critically, you do have receptors, and these are specialized cells that recognize some type of input. So for instance, when you do slap, let's say, the back of your neck, in your skin, you do have sensory receptors, you have pressure receptors, and they detect a change in pressure that is applied to the surface of the skin. Once that pressure is applied, what do you have happening? That pressure that is applied, it causes the generation of action potentials, and that action potential is then sent along the nerve to the brain where it is perceived as pain or something actually touching. All right, so in terms of the senses, Visceral ones, these detect changes in our internal environment. So visceral, that's like in particular, as it really is when you're not feeling well, right? Oh, my stomach hurting, man, right? Within your internal environment, your stomach hurting, right? Your belly hurting, you have a belly ache, right? That has to do with receptors within the stomach itself. Also, as we're well aware, they have Peyer's patches existing in the large intestine, and they also detect movements, and they give you alerts as to what is happening there. You also have somatic, widely distributed, for example, touch, and the special senses, which we'll identify shortly. So the special senses, they are sophisticated apparatus that occupy a particular or a specific anatomical site. All right. So in terms of the sequence of sensation, let's have a look at this. So here, as you quite rightly, your colleague mentioned, we have sound, we have vision, you have smell, you have taste in terms of some of these senses that we outlined here. Let's talk about touching or touch, right? So we did mention just now in terms of the mosquito coming onto your skin. So what happens there? Well, as we mentioned, you do have pressure receptors in your skin. In particular, for light touch, you have Meissner's corpuscle. And then for the heavier touch or vibrations, you have the Pacinian corpuscle. All right, so both of these they act together in terms of when you do get a touch to tell you the degree how heavy or light the touch is. Notice that all of these sensory perceptors are located within the dermis. And when we were very important to know when you think about the skin, the business end of the skin is the dermis. Yes, you have the epidermis, the subcutaneous layer with fat and also blood vessels, but the business end, where most of the structures important for the skin, where they are located within this dermis itself. Notwithstanding the Pacinian and Meissner's corpuscle, you also have free nerve endings as well as stretch receptors, the Ruffini corpuscles. So all of these things collectively help the skin in terms of letting you know what is happening around you. And as we are well aware in terms of organs in the body, the skin is the largest by surface area. If I were to ask you what organ in the body is largest by volume, what would you say? Anybody wants to hazard a guess? Which one by surface area? Sorry, by volume. What is the largest one?
So your blood.
Your blood. Blood is, you're thinking of the cardiovascular system, and I like how you're thinking because it does occupy most of the body, but there's something actually larger collectively than the cardiovascular system. But that's an interesting one, the heart and all of the blood. Now, there's something that weighs heavier by volume. So in terms, sorry, I gave you the wrong term. My bad. By mass, sorry, by mass in terms of weight. Which one is the largest in terms of by weight? By
Skin.
volume. I hear what you're saying. You might be quite right in terms of blood, the cardiovascular, in terms of the volume of blood, approximately 4 to 5 liters. So that would be correct in terms of liquid volume. You'll be quite right. But if we're talking about by weight, which organ is the heaviest?
Skin.
Anybody? Which organ is the largest one? I'll give you a hint. It rhymes with shiver.
Liver. Is it liver?
Anybody?
Liver.
All right. It is, of course, your liver. Your liver is actually your largest organ by mass. It's very large. It goes across the width of your body itself. And you will see it more so, when you do anatomy and physiology. Well, more anatomy and physiology, and specifically when you go either when you actually start your rotations, and let's say you go to, in particular, your surgical rotation, where you actually see it, or if you do look at it in terms of cadavers and so on. Okay, let's go forward. So in terms of other perceptions, you have proprioception. And what is proprioception? Well, Proprioception is that ability to tell where you are in space and in terms of where you are as related to movement. So it's not surprising that in terms of proprioception, most of the proprioception receptors are located within the muscle. Because in terms of movement, of course, when you're going from A to B, it usually involves the action of muscle. So proprioception, in terms of getting that feedback from the muscle in terms of its movement or contraction or relaxation, right? That occurs, most of the sensors are located within the muscle itself. So do take note of that. So now we go on to taste. All right, so what primary taste do we have? Sour, salty, sweet, bitter, and umami. So umami, right, is meaty or savory. Could somebody give me an example of something related to umami that gives this meaty or savory taste? What gives a meaty or savory taste? Let me
Pork.
hear you. Okay, it looks like I'll have this out of call person. Diana, what gives a nice meaty or savory taste but is actually not meat? Can you think of something? And you find it on pizzas and it rhymes with mushroom.
Mushroom. Mushroom.
You could unmute.
The
Yes,
mushroom.
Diana, yes.
Mushrooms.
Yep.
But I would have
I'm not
never know that.
hearing. I'm
Would
not
have
hearing
never.
you all.
I would have
I
never
guess
know
y'all are
that.
talking but not hearing. Okay, that would make a lot of sense. Okay. Let me see. Because I'm not hearing. So when you do speak, you'll just have to unmute yourself. And let's see how that works. Yeah. So you just have to click on the microphone when you talk to unmute
Hello?
yourself. So Diana, you want to try again? Actually, you are
Yeah.
doing it, but I'm not hearing you.
Um.
I wonder.
You hear me now, sir?
Wonder what happened. What setting? Let me. Oh, let me unmute all. Yeah. I don't know why I'm not hearing anybody. Even when Diana, I'm seeing your microphone moving, but I just not hearing you. Go figure. Okay, that is not an issue. We just we just move on from there. So the five primary tastes, they are sour, salty, sweet, bitter, and umami. And umami, that's the taste you usually get as related to um, mushrooms, right? So mushroom, that kind of taste that is associated with it is umami. And very interestingly, each of these taste sense, so in other words, then you will have receptors that detect sour, salty, sweet, bitter, and umami. They're scattered throughout your tongue. And these activate a distinct gustatory, a receptor cell. So in other words, then when you eat certain things, right, the chemicals will bind to receptors located on your tongue. When it binds to the receptor, that causes the activation of the receptor, and by extension, when the receptor is activated, it causes then the generation of an action potential. We saw when we were looking at the neuromuscular junction in terms of a synapse, when you have acetylcholine crossing that synapse and binding to the receptor, when it does so, it causes then the regeneration of an action potential on the postsynaptic membrane. So in a very similar manner, when you look at the tongue in terms of binding of these chemicals, it just, once you have this binding, it causes then this generation of this action potential. All right? Okay. So this is just showing you this diagram here. It's showing you the tongue, right? And as we, well, oftentimes you don't really see all that is located in the tongue. This is more the back area here, but you do have the fungiform, the filiform, and the valid papilla, right? Very important there. So of the three types of tongue, only the valate and some of the fungiform actually contain the taste buds. So note well, the valate and some of the fungiform. So the valate and some of the fungiform area, these actually contain. So you don't really have a lot of taste buds more to the front of your tongue. And the majority is located more to the back and close to the middle of the tongue itself. And what happens? Let's have a look. So here, for instance, what you have a taste cell, right, as located here. And when you have the binding of whatever odorant or taste to it, sodium, it causes then the opening of sodium channels. And sodium ions pass through the channel, causes a depolarization of the membrane. When you have depolarization of the membrane, this open voltage gated calcium channels, calcium rushes in, calcium then bind to these synaptic vesicles, and then these synaptic vesicles, they bind to the postsynaptic membrane, releasing chemicals, which then, of course, lead to the generation of action potentials, which then travel along the different nerve. In this case, cranial nerve 7, 9, or 10, and this goes to the area of the brain associated with gustatory or the taste sensation. So once it receives that stimulant, it identifies it by comparison to something in memory. That is very important. All right. So each of the 5 taste sense activates a different type of gustatory receptor cells. Let's try again. Could anybody remind me of the five taste sense again? What are they? The 5
Sweet,
tastes.
sour,
Okay,
is
once
it
again, I'm seeing Diane, yeah, seeing your
sour,
thing
salty,
moving, but I'm not. I'm seeing your microphone moving, but I'm just not hearing you, Diane. I wonder if it's not on my. Yeah, I'm seeing my microphone moving. Let me. I don't think. Let me just my mic to see if everything is okay. Just speaker and microphone. Huh. Increase volume to hear the button. Unmute your speaker or increase volume. Hello, testing, testing, 1, 2, 3. Hearing anything. Okay, just 1 second. Let
Hello?
me just pause this. Yeah. Hello, hello. Testing, 1, 2, 3.
Yes,
What is
sir.
going on? Hello. Hello. Yeah.
Yeah.
yes,
Y'all,
sir.
y'all were hearing me all the time?
Yeah.
Yes, sir.
Okay, excellent. All right, but now I hearing you. Yeah, I realize. Yeah, I just had to um, make an adjustment. All right, great. So I'm glad you all were hearing me. So that means it was being recording. So that is good. So let me resume share. Y'all seeing the screen again?
Yes,
Yes.
sir.
Okay. So in terms of the 5 tastings, somebody wants to remind me what they are. What are the 5 tastes? So we have what's
Sour,
that?
salty,
Sour, salty,
sweet, bitter, umami.
sweet, bitter, and umami, right? Sour, salty, sweet, bitter, and
Yes.
umami. Very good. Okay, so now we're going to smell. So on average, the human nose can distinguish more than 10,000 different odors. OMG, isn't that fascinating? And some people, of course, they do have an exceptionally high level of, they could, they could detect things very in terms of smell, you know. Anybody here have super smell by chance? Yes, no? Nobody have the super smell, but they could detect. I know I have an aunt. to have a super smell, believe you me. You know, if you cook, you could pick up something at the end of the house, you know, when you're not working, and you know, she's very, very good. Or if it is, you know, it's not well cleaned or something like that, she'll comment on it. All right. So some people do have that sense of smell that is heightened, right? Or some people have the sense of hearing, right? That is very heightened. And what do we call those people who have a heightened sense of hearing? What do we call them? Very good. We call them Makos. Very good. Yes, yes. Because they could listen in on conversation without you even knowing. Now, in all due, I just said that in jest. Please do not write that in the exam. Of people who hear very well with Makos, I will laugh and I'll give you a 0. All right? Let's go. So smell, the average human nose can distinguish among more than 10,000 different odors, and olfactory cells lose about 50% of their sensitivity in the first second, which is why, I don't know if you recognize, sometimes you will visit somebody, I know as soon as you enter the house, you might be able to detect, you know, a certain smell, but after a couple seconds or minutes, you don't smell it anymore. And it's the same thing, let's say if you're in a room and somebody comes in wearing a perfume or cologne, you would pick it up right away, but then it disappears rather quickly, you know, in that regard. Yeah, and that has to do with the fact that because these sensory cells, they lose their sensitivity within a couple of seconds, they will lose it. Now, why do you think that is important that they do that? What would happen if they didn't lose their sensitivity after a couple of seconds? What do you think would happen?
We will just be smelling the same thing.
Yeah, that would be would that be a problem? Do you think
Yeah,
that?
because we won't be able to distinguish between
Yeah, very good.
smells.
Excellent answer. Yeah, you wouldn't be able to distinguish, you know, between A and B because all the time you're getting this, you know, your nose is, let's say you walk in a room and somebody have fried chicken, so you're smelling fried chicken and that is it. And let's say now, you know, something else, you know, is being cooked. Let's say you have a pot on the fire and it burning. You wouldn't be able to pick it up or it would be very faint because of the fact you still have this sensation of the fried chicken, you're smelling it very strongly. So that is why, you know, where that is concerned, you know, you lose that sensation of smell rather quickly. All right, so here we see how it happens, the odoron. So we're talking about chemicals. Chemical binds to some of these receptors in the cribriform bone or the cribriform plate, right? And that is very close to the olfactory bulb. So what you have happening here, when they bind, they elicit or they cause the action potential to be generated within the olfactory bulb. And when that happens, it causes those action potentials to go up cranial nerve 1 up to the area of the brain associated with gustatory or the smell sensation. All right, so that is very important to take note of. Again, in terms of with smell, the generation of the action potentials occurs across the ethmoid bone by the stimulation of the olfactory bulb. Now let's we go on to this other one. So what is this sensation known as here? It's
Sound, hearing.
sound or hearing. Now, the air consists of 3 parts: the internal, middle, and external. And in the internal, here, the external, middle, and inner air. So the external here, this big thing here is, of course, is the pinna. Some people have it more pronounced than others. Here you have the auditory canal. And why do you have a pinna and an ordinary canal? Why is it shaped like this? One word. It's shaped like this to a blank sound. A word that begins with C. Or we could go with the simpler one. We could say F. 5 letters. It's a blank sound down to the tympanic membrane. Begins with F, 5 letters. And it rhymes with bocus.
Focus.
Focus, yeah. So that is, it focuses, because think about it. All the sound that's coming here is channeling it, you know, you're focusing it. And the sound now is coming down here and is able then to vibrate our tympanic membrane. And of course, when it vibrates the membrane, what happens? These 3 bones, the malleus, the incus, and the stapes, also known as the hammer, the anvil, and the stirrup, right? These 3 then begins to vibrate. A little trivia question: what is the smallest bone in the body?
Excuse me, sir. Somebody wanted to get into the class. Search in the
Okay.
group.
Mm-hmm.
Sorry.
Sure. Thank you. Ooh, let me see. Invite. No, not invite. I'm not seeing them. Okay. Thanks. Right. So the sound is focused down here, and it causes then the vibration. And then these three bones. Which one is the smallest bone you think out of these three? The malleus, the incus, or the stapes?
The steps.
Yeah. Or some people say stapes, right? That is, and little FYI, this is the smallest bone in the body. This is it, the stapes. How big is it? Well, you can look it up at your convenience, but it's really small. You know, now that I have stoked your curiosity, I'm sure you will ask me. So let me get that answer, the size, but it is really small. So let me get here. Let me define, because really small is a loose term. So let's see just how um, let's see how size of stapes bone. In terms of its size, the stapes or stapes, as it were, it's 3 millimeters by 2.5 millimeters. So what is a millimeter? As we discussed previously, if you take your thumb and your forefinger together and you press them together, right? Hold it up, let's say to a window, and start to take them apart. When you now start to see a space between your thumb and your forefinger, that's 1 millimeter. So imagine just two of those little spaces between your finger, between your thumb and forefinger. That is that's the width, and it's 3 millimeters long. This bone is so small, and yet without it, we wouldn't be able to hear a thing, all right, in terms of this little bone here, the stapes. All right, so external air, middle air, and then we have the inner air. And again, what happens, the sound comes down, it vibrates this window, which is known as the tympanic membrane. This vibrates the bones. These bones then vibrate, or they hit against the oval window. And when it does so, it causes then the movement of uh, it causes the movement of fluid within this area here, which is the inner air. And we'd speak more, a little bit more in terms of the detail. Let's just see what specifically happens within this area. Okay. Okay. So the inner air, when we look at it, this is showing the inner air. It consists of the organs of equilibrium, namely the vestibule and the semicircular canal. So this is the vestibule here. These are the semicircular canals. The membranous labyrinth is contained within the bony labyrinth, the membranous labyrinth, and is carved out of the temporal bone. So again, too, notice this structure, very important for hearing, is encased in bone within the skull itself. And we mentioned previously the importance, and when you look at important structures in the body, they're usually encased in bone. And this example, holds true to that fact that because it is encased in bone, most definitely this is important. So the next part, of course, is the cochlea, and the cross section of the cochlea is shown here. And in the cochlea, the membranous labyrinth, again, is shown here, is called the cochleal duct and contains the hair cells that detect sounds. So let me ask this question. What fluid is contained in the tympanic duct? So let's see if we could find the tympanic duct, which is here. What fluid do we have within the tympanic duct? It begins with a P.
Perilymph.
Perilymph. Yes, very good, right? So that answer will be, of course, the perilymph. Note very well, note something that's important. Both the outer and the middle air, they're involved in sound sensing, but the inner air, right? The inner air, which is what we're looking at here, this is involved in both sound sensing and equilibrium, most importantly. All right. So when it is you do have, let's say, an infection involving the inner air, you would have your equilibrium affected. Right. So some people, they'll tip over or what have you not. Okay, where that is concerned. So as I mentioned, outer and middle air, sound sensing, and the inner ear, not only does it help with sound sensing, but also in terms of equilibrium. Okay, so how is this sound? How do we perceive sound? Now, sound actually travels as waves. And these are alternating layers of compressed and decompressed air. All right, so you have some areas that compress and some that is loose. And how does this look? Well, so if you're looking at a speaker, boom, do doom, 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 do doom, 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 right? You have compressed air, and then you have decompressed areas where it's not compressed. Compress, decompress. And then this could be translated or shown in terms of the pressure, in terms of looking at difference in pressure. This compressed air will be high pressure, so this will be up here, whereas the decompressed will be low. So it could be shown then as these waves of varying high pressure and low pressure areas. Anybody ever stood up in front of a speaker at a fete, or let's say in somebody playing loud music? You ever stood up in front of a speaker? Did anybody ever did that for the duration of the night? And then when you left, heard a ringing sensation in your ear? Well, I did that.
Yeah.
I did that actually when when I was young. I remember we went to a fete in the stadium. Back then, everything was in the stadium, National Stadium. That was when all the big fete used to be. I don't remember which artist that came down, but yes, I did find myself in front of a speaker. And those, the speakers that actually taller than you, about 8 feet and so, yeah. So yes, it was enjoyable until after I left, and I hear this ringing, a ringing noise in my ear, and it took like almost a day before for it to actually go away.
And so that's what has happened to when like people go on at war and a grenade go off and there
Yes,
was no
correct.
something.
Correct, because of the high pressure wave, yeah, that comes through. Yeah, that's quite right. So in fact, there are some grenades in particular, they call them flashbang grenades. And not only do they emit a bright light to actually disorient you, but they also make a loud noise to do exactly that, to throw off your hearing. So you'll be totally disoriented. So it gives them, the attacker, an upper hand. So you're quite right in that regard. All right. So just words of the wise, don't avoid grenades in war, and also don't stand in front of a speaker. It's not very good for you. Okay, so let's talk about how hearing go through it again. So the sound wave, as we mentioned, this is the outer air, sound wave travels in, and it actually causes vibration of the tympanic membrane. By extension, the tympanic membrane then vibrates the bones of the middle ear, the stapes, the malleus incus, and the stapes. The stapes is the innermost bone, and the stapes vibrates the oval window, which is the entrance to the inner ear. So this is it here. It vibrates it, causing a wave in the vestibular and the vestibular duct perilymph, that fluid that is moving in there. This fluid movement causes a traveling wave in the vestibular, tectoral, and basilar, the membranes. Because the fluid is moving down, it causes the membrane as well to move. And when the membrane moves, the cochlear duct movement sets up a fluid wave which vibrates the round window. Right, and then when you vibrate the round window, cilia within there, so cilia, these are just here like projections, they flex as the wave passes through the tectoral and basilar membranes. So this is shown here. If you were to highlight it, the hair cell membrane potential changes when the cilia are bent. So long story short, the vibrations, what they do is they cause this movement to occur in this membrane. And when you have this movement occurring in the membrane, you have a change in the action potential. And what does that cause? It causes the release of a neurotransmitter, which then binds to the vestibulocochlear neuron. And that causes the generation of an action potential that actually travels up that nerve, cranial nerve, up to the brain, to that area where you now perceive sound, right? That perceives the sound. And again, as I mentioned previously, that sound, the only way the brain perceives the sound is as action potentials. So all of the apparatus associated with the inner, middle, and outer ear, they just serve to transmit that sound to the inner ear such that you have a deflection or a change in the membrane shape, which causes the release of neurotransmitter leading to the generation of an action potential that passes along the auditory nerve up into your brain, where it is now perceived as sound. So as we mentioned, everything is just perception in terms of your brain. You perceive the sound. The sound doesn't actually go up to the brain, but it's the action potentials that go up, and that is what is perceived in the brain itself. We don't have to, for this course, we don't have to look at the auditory pathway. For the vestibular, otolith, and otolith organs, one of the things we just have to appreciate is the fact that you do have the movement of the structures within the, again, the fluid-filled structures in the inner ear. And because you have this movement causes the generation of an action potential. So as for instance, here within the semicircular canal, when it is stretched, this causes the release of, or in this case, you have stretch receptors, sorry, and this causes the generation of action. When they're stretched, it causes the generation of action potentials that passes along the neuron itself. So those are just the takeaway message in terms of from the air. Do know that the transmission of the sound leads to the generation of action potentials that transmit via the auditory nerve up into the brain, and that's how you actually perceive the sound. Next thing we want to look at is the eye. And here it is, we have eyes. This is shown a blue eye, but of course we have different color eyes, right? We have brown, we have black, and in fact, black eyes are very rare. Most of us have dark brown eyes, but sometimes we would say black, but it's very rare. You know, just like hair color, black hair color is very rare. They're seen mostly in persons from India, of Indian origin or Chinese origin. Persons of African ancestry very rarely have black hair. It's usually a very dark brown. And one might argue it has to do with the exposure to the sun in terms of the location of Africa as it relates to the sun. And globally, you know, so you could probably well argue that. But in general, most of us, if you really look at it, we have dark brown, very dark, but black, purely black hair, only persons really, you get a lot in India and China, but you don't get it a lot, you know, from those of us who went on the diaspora out of it, you know, because we do have, you know, dilution in terms of the genetic pool associated with hair color. Okay, let's go forward. So the eye, lacrimal gland located above the eye. We have the eyebrow, the eyelids, eyelashes. We'll not be responsible for all of these detailed terms, the lateral canthus, the sclera, yes, the medial, not the canthus, no. And we just have to know the lacrimal canal, which actually drains the eye, right? But we wouldn't be looking at the canthi, the lateral and medial canthus. Now, what is the purpose of your eyebrow and your eyelashes? Anybody would want to hazard a guess? Why do you have eyebrows and eyelashes?
Oh, so I know the eyelashes is protect you from dust and stuff.
Yeah. And the eyebrow protects you from what? If you think about it, what does it protect you from? One word. 5 letters, begin with S.
Sweat.
Sweat, thank you very much. Yeah. So when you sweat, right, or perspire, some people say men sweat, but ladies perspire, right? But when you sweat, it actually does prevent the sweat from running directly down into your eyes. Of course, if you're quote unquote sweating like a horse, it wouldn't help you much, which is why some people wear headbands, you know, to absorb sweat, especially when you play sports, right? But in general, that's why your eyebrows are there for. And the eyelids, quite rightly, not only do they protect in terms of, let's say, liquids, but more so in terms of physical things actually getting into your eyes themselves. So let's have a look at the eye, you know, in terms of the structures. If you were to look at this cross section here you have the orbicularis oculi, right? So the orbicularis oculi, what is the orbicularis oculi? There's a circular muscle that goes around the eye. Very important for moving the eye around. Here's the eyelid and the eyelashes. So here's your cornea, your sclera, and of course your conjunctiva. So do take note of these structures associated with the eye itself. So these are some of the external eye muscles. Since we are looking here, We see the oblique and rectus and medial. And of course, what we'll be paying more attention to is the optic nerve, which runs from the back of the eye itself. But all of these muscles enable us to actually move the eye around. Some people do have issues in terms of with these muscles. And of course, you have different ailments or different conditions, that's the proper word, associated when these muscles, don't do their job too correctly. And what is the term used for when persons who have, let's say, their eyes are not, and usually your eyes move in tandem. So you look to the left, they both move to the left and so on. If you do have a condition where, your eyes are not moving in tandem, what is that condition known as? Anybody? In the parlance, we say, and I say this out of with no disrespect, sometimes you call it, cocky eye. But what is the correct term for it? When your eye, right? Let me see if I can pull up some images and show you. Anybody? Well, it's not on the syllabus itself, but I just wanted to bring it up. It's my the condition is strabismus. I want to say that's the word. All right. So let me see if it'll come up. You all seeing it? Yes. You all seeing this?
Yes.
Yes,
Yeah,
sir.
the different condition. Yeah, some people, they have it extreme in terms of, eyes looking in both direction. You might have one eye, going off in another direction and so on, right? So here's another example, yeah, where you don't have, and the reason why is because of the fact of those muscles. I want to say it's called strabismus, the condition itself. Strabismus. Some of the eyes don't look straight ahead, it sure is. And that has to do, very simply, with the fact that these muscles, the muscles are not, they are not synchronized with each other. So therefore, they're out of sync. Or you could have undevelopment of one of the muscles themselves so that it doesn't cause that movement to occur. Yeah. I'm sure some of you might know persons like that. And I don't know if you all, I've done a informal, I've been doing this informal like survey, and I've noticed that this condition has increased drastically, over the last couple of years. I don't know why. To me, seeing this
Is
condition,
that genetic,
yeah,
sir?
in terms of the component, yes, it does have a great genetic component to it. But the question is why, like as I say, it's an informal observation I've seen. But I've just noticed more and more, it's like, person's eye, when you look at them, like one eye a little off center. And sometimes in particular, when you look at basketball players, a couple of them, a good bit of them, they have one of the eyes off. And it's just more so, I don't know, probably if it's the increase in technology, which now allows us to see more people in terms of pictures, in terms of streaming videos and so on. But to me, it didn't used to see it. It was rather rare to see, see the condition. But more so now, you see it a lot. For those of us who follow basketball, of course, probably the most famous of them all, Shaquille O'Neal, right? He does have this condition. If you do see a straight on picture of him, you'll see one eye is off to the right and when one eye is looking straight ahead. So he does have this condition. Yeah. But anyhow, um, it does happen. All right, let's continue from current side. Okay. Okay. So this is us showing the anatomy of the eye. right? Of course, light comes in and it is focused into the, well, to the fovea itself. But in terms of the bending of the light, when it does come in, the cornea bends the light initially, the cornea, right? So it bends the light, it comes in, and then of course, ideally you'd want it goes through the lens and you want it focused on the fovea itself, all the way to the back of the eye itself. Now, interestingly, in terms of what happens to the eye when it's exposed to bright or dim light. Now, all the way to the back, the retina is very light sensitive. So you really don't want a lot of light going through to the retina itself, which is the innermost. If you were to go back, see the retina, this is the innermost layer here, the retina. So you really don't want too much light getting into it. So what happens in terms of the pupil size, which is the quote unquote hole in the eye through which the light goes, when you do have bright light, you have the circular fibers, they contract, and this causes the decrease in the size of the pupil. However, in dim light, you want as much light coming in as possible. These muscles relax, and what happens is the size of the pupil actually dilates. Now, this, of course, is under the autonomic nervous system. This happens without your knowledge itself, and it's within the brain. And one of the things that I think I mentioned it last day as well, when you do come into the ER, that's why they flicker light. If it's suspected that you did have a brain injury, they will flicker light into your eyes if you're unconscious, just to see if that pupillary reflex is still there. And if it's still there, it indicates then that that portion of the brain associated with the pupillary reflex is not damaged. All right, you could do this at your convenience in terms of this little example there, right? Okay, so let's talk about accommodation. Now, what is accommodation? Accommodation is the ability of the eye to adjust its focal length. And what do we mean by that? Somebody wants to simplify that for me? So accommodation is the ability of the eye to adjust its focal length. What does that mean? Very simply, what does it mean? Anyone? So when you're actually, let's say, sitting at a computer, or let's say you're sitting right now, home at your computer, or you're looking down at your phone, cell phone, or sitting behind a computer or a laptop, and let's say you look up and you look through a window and you're looking at a building far away, right? You're able to focus on that building. And by the same token, after focusing on the building, you could look back at the computer or your phone that's right just in front of you. And your eye actually accommodates for that change of looking distant and close. So that ability to be able to adjust the focal length of that is all it's speaking to. The ability of the eye to look at distant objects and then again to look at close objects, right? That is known as accommodation. And that is really we don't even think about it because again, too, it is done automatically. So that is really very, very important. So this is just showing accommodation, what happens for close objects. The ciliary muscle is contracted. Let me just go back here. For a distant object, the muscles are relaxed. The ciliary ones, the ciliary body is flattened, shown here. And the suspensory ligaments that actually hold the lens in place, they are taut. So what happens? So they're pulling. Think about it, that these ligaments, they're pulling on the lens, so the lens is flattened, and that's allows you to see far away. However, for something close, these suspensory ligaments are relaxed, so the lens now become more rounded, so it's now able to bend or refract the light more, such that you could form that image on your fovea. So this is shown here, right, in terms of accommodation for close objects, and this is for a distant object. All right, so two major, when you look at the eye, the major photoreceptors within the eye itself, they are the rods and cones. And the rods, these are primarily in the periphery, and the cones, they're primarily in the fovea centralis, so the middle part of the eye. They're cone-like in shape, and the cones, as C is for cone and C is for color. Cones, they sense color and fine detail, whereas rods discriminate light from dark. So rods, you use them more at night, let's say when there's not a lot of light available, but cones, they use them particularly, lots of them, during the course of the day itself. So this is just showing, and what happens interestingly, once they are stimulated, again, they cause the generation of these action potentials. So if the color ones are generated, right, you have a particular image which can now be formed in the brain itself. But these images are nothing more than because of the fact that you have the stimulation of these particular cones, these color photoreceptors associated with the brain itself. Of course, you have three major ones, red, green, and blue. And depending on how many of these are stimulated, you could have a combination of red, green, and blue to give virtually all the known colors which we actually perceive. Of course, if you have no stimulation of these receptors, what color would you see? If you have no stimulation, what color would you see? Anybody? If you have no simulation, what color would you see? If you had to make a guess, what color do you think it is?
Black and white.
Excellent. You would see black. So black is really the absence of color, whereas when you think about all the colors being blended together in equal proportions, you will get white. So could you, it's probably best to try this with light as opposed with pigments in terms of paints, because paints are really not pure. You don't really have pure. They come pretty close to red, yellow, red, green, and blue, right? But they're not actually pure. So you'd come close to white if you mix them in equal proportions and red, green, and blue, but probably get more of a kind of a gray because they're not truly pure. You really get the combination to give a white. When you overlap these three, you get more with, if you have filters that give pure colors of red, blue, and green. Some color monitors as well, because of this fact, they use red, green, and blue to generate virtually all of these colors that we see. All right, so in terms of cones, as we mentioned, they provide higher image resolutions. So a cone associated with colors, and of course, because of the fact you could differentiate more colors, they're more defined. So cones are associated with better image resolution. And in presence of light, right, these rods hyperpolarize. And in terms of hyperpolarize, it means that in the presence of light causes a change to occur, ultimately leading to the generation of action potentials. So that's just the take-home message, especially with all of these senses, is the generation at the end of it all is must have a generation of an action potential. So with the air, you have differentials in pressure, but what does it do? It passes from the outer to the middle and then to the inner air, where you have the transfer to the oval window along that, causing distortion in the membrane, and the distortion in the membrane causes then generation of action potential, because when you do have the distortion, you do have the release of certain, you will ultimately have the release of an action potential, which then travels along the auditory nerve. Similarly, when we smell things, you do have these odorants or chemicals which are released. They bind to specific receptors, and then they cause because of the binding, you do have probably G-coupled receptors. When you have binding to G-coupled receptors, you do have the generation of action potentials occurring subsequently, right? So all of these things with light receptors. So here, in terms of light, is a special case, and it shows you here, but ultimately what you have is the stimulatory neurotransmitter causes a depolarization and it fires an action potential. So that's the important take-home message there. So important to note as well, neurons from the medial side of the eye, they cross over in the optic chiasm. Neurons from the lateral side do not. I mentioned this, but this is more for your edification. It's not on the syllabus in terms of knowledge of the optic chiasma. All right. So we just mentioned here, let's have a look at this. So let me just give, in terms of this, if I take home, let me just give you a question here. So this is just showing sense, stimulus, sensory organ, and so on. So let me give you a scenario. Helen's sensory organs and receptors were, in all likelihood, intact. However, signals originating in the hair cells of the cochlea and the photoreceptors of the retina failed to reach the brain. Taste, which was not affected by Helen's disorder, is not illustrated. Note that the pathways are simplified. So the question is, name 3 stimuli that activate hair cells. So which 3 hair cells activate, which 3 stimuli activate hair cells? So which ones activate the hair cells? So we are looking at in the air associated with hearing. Which stimuli activate hair cells?
So,
Yep. What would you say? What could activate it associated with the air?
you mean sound, like sound entering
Sound.
the
Yes, sound is
ear.
one. Very good. Sound. There's another one. Related to sound, you could have acceleration because, in terms of spinning, like if you're a ballerina. So this is it shown up here, right? Acceleration. And of course, gravity, right? When you move your head up and down, they also cause the stimulation. All right, so that's the end. Thanks very much for that response there. That's very brave of you. So that's the end of the presentation today. One thing so we looked at the 5 senses, which is sight, smell, taste, hearing, and of course, touch. Those are the major ones which we looked at. Very important to take note of with all of these. Do remember, you don't have a tube, let's say from your nose straight up to your brain, but instead, with all of these senses, what is very critical to remember is that ultimately the binding of, in the case of smell, odorant chemicals, in the case of taste, when you have molecules actually from the food actually binding to the specific receptors located in the tongue. And as we mentioned, you have the different specific ones related to sweet, salty, bitter, umami. Did I forget any? Sweet, salty, bitter, umami. Those are the 4. I don't think I left out any. But when they bind, they ultimately cause generation of action potentials. Similarly, when you look at the photoreceptors in your eye, you do have, in terms of hyperpolarization and hypopolarization of the different within the rods and the cones. And this leads to the generation of action potentials, which are then sent to the visual cortex located to the back of the brain, right? Very important there. So all of these sensations, right? The smell, touch, hearing, equilibrium, and vision, smell, touch, hearing, taste, and vision. The end result of all of them, when you have the binding of a sensory perception in the case of sight and hearing, and specific odorant molecules in the case of taste and smell. And then, of course, well, for the perception, you also have pressure, changes in pressure in terms of sensation that is received in the skin itself. But ultimately, just the take-home message from this is that they all lead to the generation of action potentials, which travel along the cranial nerves to the brain. And it is within the brain, the differences in the stimulations are perceived, and then by a means of comparison to memory, that is how you're able to identify, be it a smell, a particular touch, hearing, what do you hear, and what do you see, is because of this comparison. Okay? Any questions? All right, so we'd stop here for today. Let me see.